Welcome to the Bonito Lab training video on PCR cleanup with XOAP and Sanger sequencing submission. I'm Julian Liber. First, you will need additional PCR strips and lids. Pour out the number you need to avoid contaminating the stock. Seal bags carefully after use. Carefully label the strips you will use. There are numbers on the tubes which you can use to keep the orientation correct. We will use 1.2 microliters XOAP per sequencing reaction. XOAP master mix is a combination of two enzymes, exonuclease 1 and antarctic phosphatase, in the appropriate buffer. This mix is viscous and difficult to pipette, so watch that the liquid ends up in the tube. XOAP cleans and modifies the PCR product to be ready for Sanger sequencing. You should be able to see the master mix in the bottom of the tube. Now we can add the PCR product, 1.5 microliters per sequencing reaction. Centrifuge strips prior to carefully opening the caps. You should only attempt to sequence PCR products that showed strong bands in the correct fragment size with gel electrophoresis. Check that the PCR product ended up in the well and is not stuck in the tips. If so, try pipetting up and down, or get new tips and try again. Be careful to carefully map out the XOP reactions to keep track of the sample, because you may be skipping wells or using multiple PCRs. If you are sequencing your products with more than one primer, you can multiply the XOAP master mix and PCR product amounts. Label your strips and centrifuge them before placing on the thermocycler. Set the cycle to react for 30 minutes at 37 degrees C, then 15 minutes at 80 degrees C, and 10 C until the reactions are removed. The minimum sample volume is 5, so use that. Clean up your workspace while the samples are on the thermocycler. After the reaction is finished, remove your samples and place on a microplate rack. You will need to have prepared a mixture of 0.2 microliters of one primer and 10.8 microliters of water for each reaction. you should do this in the pre-PCR hood. You can only use a primer which is complementary to your PCR fragment at the ends or internal. Add 1 microliter of XOAP product to your primer water mix. Place strips in the rack, rotating to make the first reaction in the orientation designated on the submission form. This is standard microplate positioning. Tape your strips to the disposable rack with a descriptive plate label. This will also go on the submission form. Fill out the Sanger sequencing sample submission form on the RTSF Genomics website, giving the relevant information for your submission. You will check the client box under primer. If you are a new user, you will be contacted by the facility staff. Follow their protocols on where to drop off samples.